Welcome to the second session on human reproduction. In this session, we will mainly discuss the topics like gametogenesis and menstrual cycle. You know what is gametogenesis? Gametogenesis is the formation of the male gamete and the female gamete, and it is formed by the corresponding primary reproductive organs. In the case of male, testes, they are the primary reproductive organs, and it will produce sperm cells, and in the case of females, ovaries will produce ova. So the process of forming these two types of gametes is known as gametogenesis. There are different terms for these two processes. Formation of sperm cells is known as spermatogenesis, and formation of ova is known as oogenesis. So spermatogenesis and oogenesis, these are the two types of gametogenesis. First, we will discuss spermatogenesis. Spermatogenesis in a male individual will begin when he attains puberty. When the individual attains puberty, there will be increased secretion of hormones. Particularly, hypothalamus will produce gonadotropin releasing hormone, and that will be affecting on anterior pituitary, and as a result, anterior pituitary will produce the corresponding hormones. If you start spermatogenesis from spermatogonia, you can see here in this diagram, spermatogonia, they are the cells from which sperm cells are produced. These spermatogonia, they are diploid cells. You know what is a diploid cell? In a diploid cell, there will be two sets of chromosomes. So here, spermatogonia, they are with two sets of chromosomes, or they are diploid. These spermatogonia, they are undergoing repeated mitotic divisions. And these repeated mitotic divisions will lead to formation of more and more spermatogonia. And the process is called phase of multiplication. So the first phase in spermatogenesis is phase of multiplication. So as a result of phase of multiplication, the number of spermatogonia will be increased. And what happens next? Each spermatogonium, singular term is spermatogonium, as you know, each spermatogonium will undergo a growth phase. And when it undergoes a growth phase, there will be formation of primary spermatocytes. So the second phase here in this diagram, you can see this red colored cell is spermatogonium, and it is transformed into a bluish colored cell here, that is primary spermatocyte. And this process is called phase of growth. So after phase of multiplication, there is phase of growth. Phase of growth results in primary spermatocytes. Actually, these primary spermatocytes are the cells which undergo meiotic division or reduction division. What is meiotic division or reduction division? In a meiotic division or in a reduction division, the number of chromosomes will be halved. Here, the diploid cell, the primary spermatocyte, will undergo meiotic division, and now the chromosome number will become half, or the cells formed from here will be haploid cells. In this meiotic division, there are two phases or two stages, that is meiosis 1 and meiosis 2. In meiosis 1, the cells formed here, they are secondary spermatocytes. And after meiosis 2, the second meiotic division, from each secondary spermatocyte, there will be formation of two spermatids. So from a single primary spermatocyte, 4 spermatids will be formed. It is important to note here that the secondary spermatocytes as well as the spermatids are haploid, or their chromosome number will be 23. So from each primary spermatocyte, the result will be 4 spermatids. Now what happens next? These spermatids, they are not functional. To be functional, they should be differentiated, or there will be some modifications happening in spermatids. And this differentiation or transformation of spermatids into functional sperms or spermatozoa, that is called spermiogenesis. So this conversion is called spermiogenesis. Okay. So there are 3 phases here in spermatogenesis. As I told you in the beginning, first is phase of multiplication, second is phase of growth, and the third is phase of maturation. By phase of maturation, the primary spermatocyte is undergoing reduction division, and there will be a result of spermatids. And spermatids, they further undergo a differentiation, or they become functional to form spermatozoa, and that process, as I told you, that is spermiogenesis. And after the formation of the sperm cells within seminiferous tubules, these sperm cells, after becoming functional, they will be released out, and this process is called spermiation. So what is spermiation? Spermiation is the release of sperm cells or spermatozoa from seminiferous tubules. So after spermiogenesis, already we can see all the sperm cells with their head region embedded in the Sertoli cells, and after this stage actually you can see spermiation happening. So now you are clear with what the process of spermatogenesis. Here in this slide you can see the hormonal influence of spermatogenesis. As I mentioned earlier, the entire process of spermatogenesis will begin only when individual attains puberty. When individual attains puberty, there will be an increased production of GnRH. What is GnRH? Gonadotropin releasing hormone. And this gonadotropin releasing hormone will act on anterior pituitary. Then what happens? You can see here in this flow chart, the anterior pituitary will produce 2 sets of hormones. One is FSH and other is LH. FSH is follicle stimulating hormone. What is LH? LH is luteinizing hormone. Here the FSH or follicle stimulating hormone in the case of male will act on Sertoli cells, and when Sertoli cells are stimulated, what happens? It will produce some factors that are essential for spermiogenesis. As I told you, spermiogenesis is a conversion of spermatids into spermatozoa. So that will be assisted by these Sertoli cells. Now what about the LH? LH, which is acting on Leydig cells, it will stimulate the synthesis of androgens. I told you in the last session that Leydig cells are involved in production of androgens. A major hormone is testosterone. So here androgens are secreted by the influence of LH on Leydig cells. So the entire process of spermatogenesis is being controlled by a set of hormones as shown in this diagram. And as I mentioned, this entire process will begin only when the individual attains puberty. In the case of male, spermatogenesis will begin only when the individual attains puberty. It is different from that of a female, that we will discuss later. Now we are going to analyze the structure of the sperm cell. In this slide you can see a sperm cell with different regions. First of all, there is a head, a neck, then this is the neck region, below that there is a middle piece, then there is a tail portion. So head, neck, middle piece and tail, these are the different regions of a sperm cell. Within the head region we can see two main structures. What are they? You can see a haploid nucleus, and this nucleus will contain the chromosome, the genetic material, and you can expect it will be haploid or it will have 23 chromosomes in it. So a haploid nucleus is located in the head region. Just above that, here in this diagram you can see an acrosome. You can see that acrosome. There is a conical structure and it is formed from Golgi complex. A cell organelle called Golgi complex will result in the formation of acrosome. And acrosome is filled with a set of enzymes. A set of enzymes which will help in penetration of the sperm cell through the egg layers. The egg cell is having different layers over it. So to penetrate through these different layers present on the egg surface, a set of enzymes are necessary, and all these enzymes are located in acrosome. And acrosome, this collection of enzymes within it, we can call it as sperm lysins. Sperm lysins. Sperm lysins are of three types. You know them: hyaluronidase, corona penetrating enzyme, zona lysin. So there are different types of enzymes located within acrosome. So all these enzymes will help in what? Penetration. Now in the neck region, what we can see? In the neck region we can see centrioles. There is a proximal centriole as well as a distal centriole located in the neck region. Of them, the proximal centriole will help in cleavage. It will form, what is cleavage? It is a repeated mitotic divisions happening in a zygote. So it is helping in the process of cell division by forming spindle fibers. So neck region is having proximal centriole, and also there is a distal centriole which will form an axial filament. It is acting as a basal body to form the axial filament. Now in the middle piece there is a set of mitochondria which will provide the energy for the movement of the sperm cell. So mitochondrial spiral we can see in the middle piece. They are arranged in a spiral manner. Then of course the tail portion, it is nothing but a flagellum which will help in the easy movement of the sperm cell. A male individual will produce around 200 to 300 million sperm cells in one ejaculation. And to call a male fertile, at least 60% of these sperm cells should have normal size and shape, and at least 40% of them should have vigorous motility also. So then only we can call the male individual as a fertile one. So the quantity is more important here. Now coming to oogenesis. What is oogenesis? As you know, oogenesis is the formation of the ova or ovum, singular ovum. Oogenesis is very much different from spermatogenesis because oogenesis will be initiated during the fetal life itself or during the embryonic development itself. Oogenesis will start. And no more oogonia. You know the cell from which oogenesis will begin, that is oogonia or oogonium. No more oogonia will be added after birth of an individual. All the oogonia that has to be formed in a female will be developed already during the fetal development, and no more oogonia will be added after birth. That is a very important point, you have to note that. Now coming to this diagram or this flow chart, here you can see oogenesis, which is the starting cell here, oogonia. And again, these cells are also diploid. And the first phase, you know, what is first phase? The phase of multiplication. As in spermatogenesis, here also we can see the phase of multiplication. The oogonia, they are undergoing repeated mitotic divisions, and it results in more and more oogonia. And after this, you can see these oogonia developing into primary oocytes. Each oogonium will develop into a primary oocyte by a phase of growth. So the second phase here, that is phase of growth. From this red colored cell, the red colored cell is oogonium. From that oogonium, this bluish colored cell is formed, that is primary oocyte. So that conversion is called the phase of growth here. And after that, what happens? All these primary oocytes, they will begin the first meiotic division, but they are all getting arrested in a phase called prophase 1. All the primary oocytes, they will begin meiosis, and they are all getting arrested in a phase called prophase 1. And further restart from the prophase 1 will happen only when the individual attains puberty. When an individual attains puberty, one by one, each primary oocyte will undergo the first meiotic division. But here, the first meiotic division will be unequal. The primary oocyte will develop into a secondary oocyte, and at the same time, a first polar body. You can see in the diagram. The primary oocyte is converted into a secondary oocyte and a small polar body. In that polar body, there is no cytoplasm, only the nuclear content is there. Now what happens next? These secondary oocytes, they will further enter into the second meiotic division. And the second meiotic division will begin only, it will not complete. It is not getting completed until the sperm enters. So again there is a phase of arrest. The secondary oocyte will be arrested, the cell cycle will be arrested in metaphase 2. So in metaphase 2, the secondary oocytes, their cell cycle is arrested. And the secondary oocytes, they further develop into an ovum, or there will be completion of the second meiotic division only when the sperm enters into it. So here in this diagram you can clearly see the secondary oocyte undergoing second meiotic division, and again you can see the second meiotic division is unequal. There is formation of a single ovum and a second polar body. So here it is again unequal. So as a result of oogenesis, you can see from one primary oocyte there is only formation of one ovum. When compared with spermatogenesis, in which primary spermatocyte will give rise to 4 sperm cells, here the primary oocyte will give rise to only one ovum. So that is a major difference here. Another difference is the unequal division happening here. Unequal because the first meiotic division will result in a first polar body and a secondary oocyte, and second meiotic division will result in an ovum and a second polar body. So the division is unequal and there is formation of polar bodies here. So there will be a lot of differences between spermatogenesis and oogenesis. You know, the entire process of oogenesis is actually happening within the ovary, and within ovary you can see all these primary oocytes that are formed from oogonia will be surrounded by a set of granulosa cells, and then we can call it as a primary follicle. So in this diagram you can see a primary follicle here. Within the primary follicle, cell will be primary oocyte, and that primary oocyte, as I told you, it is arrested temporarily in a stage of called prophase 1. Prophase 1. So primary follicle is formed by covering of granulosa cells. The primary oocytes, when covered by a layer of granulosa cells within ovary, we can call it as a primary follicle. Then what happens? Next slide you can see the primary follicle is getting converted into a secondary follicle. What is the difference here? The difference is there will be accumulation or formation of more layers of granulosa cells, and also there is formation of a layer called theca. So when more granulosa cells are added and a new theca layer is formed, then we can call it as a secondary follicle. Further, this secondary follicle is getting transformed into a tertiary follicle. You can see that here in this diagram. It is getting converted into a tertiary follicle. What is the difference between tertiary follicle and secondary follicle? What is the modification here? Within tertiary follicle, you can see a development of a cavity, and that cavity is called andrum. It is a fluid-filled cavity. It is filled with a fluid called liquor folliculae. And also, the theca layer which was there in secondary follicle will organize into an outer theca externa and theca interna. So, the theca layer undergoes a change. It is organized into theca externa and theca interna, and also within the follicle there is development of a cavity called andrum. So now in this stage we call it as a tertiary follicle. In the tertiary follicle, in the beginning also there is primary oocyte. But what happens to this primary oocyte? This primary oocyte will undergo the completion of the first meiosis, and it is getting transformed into the secondary oocyte, and there will be liberation of first polar body here. All these changes from primary to secondary, from secondary to tertiary, all these changes are happening under the influence of a hormone. You know that hormone, that is follicle stimulating hormone. So follicle stimulating hormone is helping in this transformation of follicles from primary to secondary, from secondary to tertiary, and a fully developed follicle, fully developed tertiary follicle is known as Graafian follicle.
നൂറ്റാണ്ടുകളായി ഭാരതീയർ രോഗങ്ങൾ തടയുന്നതിനും രോഗചികിത്സയ്ക്കും ഉപയോഗിച്ചു വരുന്ന ശാസ്ത്രമാണ് ആയുർവേദം. ആയുസ്സിനെ പറ്റിയുള്ള അറിവാണ് ആയുർവേദം. പരമ്പരാഗതമായി നാം ആയുർവേദം സ്വീകരിച്ചു വരുന്നുണ്ട്. ഇപ്പോൾ മോഡേൺ മെഡിസിൻ ഉണ്ടെങ്കിലും ആയുർവേദം ധാരാളം സാധ്യതകൾ ഉയർത്തിക്കാട്ടുന്നുണ്ട്. ഇപ്പോൾ ടൂറിസവുമായിട്ട് ബന്ധിച്ച് പലരും പറയുന്നുണ്ട്. ശരിയാണ്, ടൂറിസവുമായിട്ട് ബന്ധിച്ച് ആയുർവേദക്കാർക്ക്, ആയുർവേദം പഠിച്ചവർക്ക്, ആയുർവേദ വൈദ്യന്മാർക്ക് അവർക്കൊക്കെ സാധ്യതയുണ്ട്, ശരിയാണ്. അതല്ലെങ്കിൽ തന്നെയും നമ്മുടെ മനുഷ്യ ശരീരത്തെ പറ്റിയുള്ള പഠനം, രോഗങ്ങൾ വരാനുള്ള കാരണങ്ങൾ, രോഗചികിത്സയുടെ രീതികൾ, ഇതെല്ലാം ആയുർവേദത്തിലും ധാരാളമായിട്ടുണ്ട്. അപ്പോൾ നമ്മൾ പല വിഷയങ്ങളും പഠിക്കേണ്ടി വരും. ഇപ്പോൾ ആയുർവേദത്തിൽ പോകുമ്പോൾ സംസ്കൃത ഭാഷയുമായിട്ട് അല്പം ബന്ധം വേണം മനസ്സിൽ വെച്ചുകൊള്ളണം. കാരണം അടിസ്ഥാന ഗ്രന്ഥങ്ങളെല്ലാം സംസ്കൃതത്തിലാണ് കിടക്കുന്നത്. അഷ്ടാംഗ ഹൃദയമായാലും മറ്റ് പുസ്തകങ്ങൾ എല്ലാം സംസ്കൃത ഭാഷയിലാണ്. ഇപ്പോൾ കായ ചികിത്സ, നമ്മൾ ഈ മോഡേൺ മെഡിസിനിൽ പറയുന്നതിനൊക്കെ സമാന്തരമായിട്ട് ഇവിടെ ആ ശാഖകളെല്ലാം ഉണ്ട്. ഇപ്പോൾ കായ ചികിത്സ ഇന്റേണൽ മെഡിസിൻ എന്ന് പറയുന്നതാണ്. അല്ലെ ശല്യതന്ത്രം സർജറിയാണ്, ശാലാക്യതന്ത്രം, ഭൈഷജ്യ കൽപ്പന, ദ്രവ്യഗുണ വിജ്ഞാനം, മെറ്റീരിയ മെഡിക്കയാണത്. സ്വസ്ഥവൃത്തം എന്ന് പറയുന്നത് പ്രിവെൻറ്റീവ് മെഡിസിനാണ്. പ്രസൂതിതന്ത്രം ആൻഡ് സ്ത്രീരോഗം, ഒബ്സ്റ്ററ്റിക്സ് ആൻഡ് ഗൈനക്കോളജി. കൗമാരഭൃത്യം, പീഡിയാട്രിക്സ് എന്ന് പറയും. ആയുർവേദ സിദ്ധാന്ത ആൻഡ് ദർശന, ബേസിക് പ്രിൻസിപ്പിൾസ്. പഞ്ചകർമ്മമുണ്ട്, മാനസിക രോഗമുണ്ട്. ആഗതതന്ത്രം ടോക്സിക്കോളജിയാണ്, വിഷങ്ങളെപ്പറ്റിയുള്ള പഠനമാണ്. ക്രിയാശാരീരം ഫിസിയോളജിയാണ്. രോഗനിദാനം വികൃതി വിജ്ഞാനം എന്ന് പറയുന്നത് പത്തോളജിയാണ്. അപ്പോൾ ഇങ്ങനെയുള്ള പല വിഷയങ്ങളും പഠിക്കേണ്ടി വരും, സംസ്കൃതവും പഠിക്കേണ്ടി വരും. അപ്പോൾ ഇത് കഴിഞ്ഞ് ഈ ആയുർവേദത്തിലുള്ള ബിരുദം BAMS ആണ് എൻ്റെ ബിരുദത്തിൻ്റെ പേര്, ബാച്ചലർ ഓഫ് ആയുർവേദിക് മെഡിസിൻ ആൻഡ് സർജറി. അത് കഴിഞ്ഞ് അതിന് ഉപരിപഠനം MDക്കും സാധ്യതയുണ്ട്. ആയുർവേദം പഠിച്ചു കഴിഞ്ഞാൽ തീർച്ചയായിട്ടും ആധുനിക കാലത്ത് ആയുർവേദം വളരെ വളർച്ചയുണ്ട്. പാശ്ചാത്യ രാജ്യങ്ങളിൽ പോലും കൂടുതൽ സ്വീകാര്യത വന്നിട്ടുണ്ട്. അമേരിക്കയിൽ പോലും ആയുർവേദ കോഴ്സുകൾ ഇപ്പോൾ നടക്കുന്നുണ്ട്. അപ്പോൾ ആയുർവേദം പഠിച്ചാൽ തീർച്ചയായിട്ടും ഇത് പ്രാക്ടീസ് ചെയ്യാം. എന്നുമല്ല, ലോകത്തിൽ പല രാജ്യങ്ങളിലുള്ള ആളുകൾ കേരളത്തിൽ വന്ന് ആയുർവേദത്തിന് വരുന്നത് നമുക്കറിയാം. അപ്പോൾ അവർക്ക് വേണ്ട ഈ ചികിത്സയ്ക്കുള്ള സൗകര്യങ്ങൾ നമ്മൾ ഇനിയും കുറേ കൂടെ വർദ്ധിപ്പിക്കേണ്ടിയിരിക്കുന്നു. നിലവിലുള്ള സൗകര്യങ്ങൾ അപര്യാപ്തമാണെന്ന് കണ്ടിരിക്കുന്നു. അപ്പോൾ ടൂറിസവുമായിട്ട് ബന്ധപ്പെട്ട് മാത്രമല്ല, നമ്മുടെ നാട്ടിലെ സാധാരണ ചികിത്സയായാലും ടൂറിസ്റ്റുകളുടെ ആയാലും നമുക്ക് ധാരാളം സാധ്യതയുണ്ട്. പിന്നെ ഗവൺമെൻറ് ജോലിയുണ്ട്, സ്വകാര്യ ആശുപത്രികളുണ്ട്, ഹെൽത്ത് കെയർ സെൻ്ററുകളിലൊക്കെ പോകാം. പിന്നെ പലപ്പോഴും റിജുവനേഷൻ എന്ന് പറയും, എല്ലാ കൊല്ലം പോയി ഞാൻ പിഴിച്ചിൽ നടത്തുന്നൊക്കെ പറയുന്നുണ്ട്. ആ തരത്തിലുള്ള ചികിത്സകൾ സാധ്യതയുണ്ട്. സ്വകാര്യ പ്രാക്ടീസ് ആകാം. ഈ കാര്യങ്ങളൊക്കെ മനസ്സിൽ വെച്ച് കഴിഞ്ഞാൽ നമുക്ക് പോകാവുന്ന നല്ലൊരു പഠന മാർഗമാണ് ആയുർവേദം.
Now we can go to the next portion, that is menstrual cycle. What is menstrual cycle? Menstrual cycle is the reproductive cycle happening in female primates. In the case of non-primates, the reproductive cycle is known as estrous cycle. In the case of primates like man, monkey, apes, we are all primates, and in primates we can see this menstrual cycle, the reproductive cycle, and usually it is having a duration of 28 days, but you know it will vary according to the physiological condition of the person, and also it will differ according to individual. But we are taking it as 28-day cycle in the usual case. The first menstruation in a female is called menarche. Menarche, it will happen when the individual attains puberty. And the menstrual cycle, it is a cyclic process, and it will continue up to 45 or 50 years of age. Menstrual cycle, here in this diagram you can see it has got 3 phases in it. What are the 3 phases? The first phase is called menstrual phase. Menstrual phase will last for 3 to 5 days. So from day 1 to day 5 we can expect the menstrual phase or the bleeding phase. Followed by that there will be a proliferative phase. Proliferative phase is from day 6 to day 14. On the 14th day, if it is a 28-day cycle, on the 14th day or in the middle of this cycle there will be ovulation, or you can call it as an ovulatory phase, and after that there is a secretory phase. From 15 to 28 there is secretory phase. So in this diagram you can clearly see all these phases. You know, the cycle of menstrual cycle, it is happening within ovary, And also there are a lot of changes happening in the uterus, on the wall of the uterus. Here you can see the uterine events. From day 1 to day 5, you can see it is considered as a bleeding phase, or we can call it as a menses. Then from 6 to 14, in this diagram you can see there is thickening of the endometrium. The endometrium, that is the inner wall of the uterus, it is undergoing thickening, and the thickening period or that phase is called the proliferative phase. And here 15 to 28, we can call it as the secretory phase, because during this phase the endometrial wall will have very active tubular glands on it. So these are the uterine events shown here. So according to uterine events, we can call this portion from day 6 to 14 as proliferative phase, and from day 15 to 28 as secretory phase. But with respect to ovary, with respect to changes happening in the follicle, we can call the same proliferative phase as a follicular phase, because during this period the primary follicle is getting transformed into a secondary follicle, and the secondary follicle is getting transformed into a tertiary follicle. So all these changes happening in the follicle, with respect to that, we can call it as a follicular phase. And you can see exactly in the middle of the cycle what is happening. The fully matured tertiary follicle or the Graafian follicle, it is undergoing a rupturing, and there is release of a secondary oocyte, that is called ovulation. And after that you can see the ruptured Graafian follicle is transforming into a yellowish structure. What is that yellowish structure called? It is called corpus luteum. And now corpus luteum is functional, or it is beginning to produce a hormone called progesterone. That is why this phase is also called luteal phase. So with respect to ovary, from 15 to 28, that period can be called luteal phase, because corpus luteum is functional. And you can further see that corpus luteum is undergoing a regression, or it will undergo degeneration, and it is getting transformed into a corpus albicans. Corpus albicans, it is a whitish structure. So in this diagram you can see all these phases happening in uterus and also happening inside the ovary. All the ovarian events and uterine events are shown here. You know, these events are being influenced by pituitary hormones. The level of pituitary hormones will have a major influence on all these phases. During the proliferative phase, you can see there is an increased secretion of FSH. The FSH as well as LH will gradually increase during the follicular phase, and it will stimulate the development and secretion of estrogens by the growing follicles. And you can also see the LH and FSH attaining a peak level in the middle of the cycle, and this will happen around the 14th day of the menstrual cycle. And this peak level of LH, we can call it as an LH surge. Actually, this LH surge is responsible for ovulation. The growth of follicles, it is done by FSH, but the rupturing of the follicle, rupturing of the Graafian follicle and release of the secondary oocyte, it is being done by LH here. So LH, or luteinizing hormone, that is the hormone having a major influence on ovulation. In this diagram you can see the influence of ovarian hormones. As I told you, when follicles are developing, these follicles will release estrogen. So the level of estrogen will continue to increase during the proliferative phase. And further, during the secretory phase, we can see the progesterone level is increasing. Why progesterone level is increasing during the secretory phase? It is because corpus luteum is functional. Corpus luteum is producing increased amounts of progesterone. That is why progesterone level seems to be very high during the secretory phase. So in this diagram you can see estrogen being high during the proliferative phase, whereas progesterone labeled as a red line here being very high during the secretory phase. You know, this progesterone is very essential for maintenance of pregnancy as well as for implantation. So that is why this progesterone is also called the pregnancy hormone. During pregnancy, you might have noticed the menstrual cycle will be stopped and there will be no menstruation during pregnancy. In the absence of fertilization, you can also notice that corpus luteum will degenerate. Actually, the withdrawal of progesterone is responsible for the beginning of the next cycle. The next menstrual cycle will begin due to the withdrawal or decreased level of progesterone due to the disintegration of corpus luteum. In human beings, you know, menstrual cycle will happen from menarche to menopause. Actually, the reproductive years of a female is supposed to be from menarche to menopause. From menarche to menopause, they are reproductively functional. Cyclic menstruation is an indicator of normal reproductive phase in an individual, or we can say that that individual is reproductively healthy. Now we can answer some questions. Here is your first question. In spermatogenesis, the phase of maturation involves. We have a set of options. Option A, growth of spermatogonia into spermatocytes, formation of spermatogonia from gonocytes through mitosis. C, formation of spermatids from primary or secondary spermatocytes through meiosis. Option D, formation of spermatozoa from spermatids through meiosis. And option E here, formation of primary spermatocytes from spermatogonia. Can you answer this question? You know, spermatogenesis has got three phases: phase of multiplication, phase of growth, and phase of maturation. Phase of multiplication is formation of spermatogonia by repeated mitotic divisions. Phase of growth, you know, that is formation of primary spermatocyte from spermatogonium. Here the question is about phase of maturation. You know, phase of maturation is the reduction division or meiotic division. Which cell undergoes meiosis in spermatogenesis? You know that. It is primary spermatocyte. So, the conversion of the primary spermatocyte or the secondary spermatocyte into spermatid is known as phase of maturation. So, correct answer for this question will be option C. Second question. Select the correct sequence of events in the development of follicles in ovary. We have some events here. Event 1 or event P, primary oocyte within the tertiary follicle grows in size and completes its first meiotic division. Then second event, tertiary follicle changes into mature follicle and or it is also called the Graafian follicle. Then the next event given here is release of secondary oocyte or ovum from the ovary by a process called ovulation. As you know, these events are actually given in the correct sequence here. So the correct answer will be P, Q, R. So you can look for our options. Yes, our answer will be option D now. So the correct answer is option D because all the events are given in the correct order. Now coming to question number 3. Choose the correct combination of labeling for the hormonal control of female reproductive system. Here we have that diagram. So some structures or some hormones are being labeled as A, B, C, D. So you have to find out the correct labeling from the set of options. You know, for this question, you know what is A here? The A will be a hormone which is released from hypothalamus and acting on anterior pituitary. And you can guess it is gonadotropin releasing hormone or GnRH. What is B here? That will be a set of hormones being secreted from pituitary and acting on ovary. You can guess, or you know that it is FSH and LH. What is C here? Something being released from ovary. You know that from ovary, under the influence of FSH and LH, what will be released? There will be release of ovarian hormones. You know what are the different ovarian hormones? Estrogen and progesterone. Yes. So C will be estrogen or progesterone. And what about D here? That is uterus. All these ovarian hormones are acting on uterus. So our answer will be option B here. Option B. Now coming to question number 4. The secondary oocyte is liberated from the ovary under the influence of LH after completing. Here we have a set of options and you have to pick the correct one. You know, the secondary oocyte is formed after meiosis 1. The primary oocyte, when it undergoes meiosis, there will be formation of a secondary oocyte and liberation of a first polar body. And this will happen inside the ovary or inside the tertiary follicle. So after meiosis and also after liberation of the first polar body, a secondary oocyte is formed. And actually this secondary oocyte is being released from a Graafian follicle. Under the influence of LH, as I told you, the Graafian follicle will rupture and it will release the secondary oocyte. So the secondary oocyte will be released after completion of meiosis 1 and after liberation of the first polar body. So answer will be option D here. What about meiosis 2? You know, meiosis 2 will happen only when the sperm enters into the liberated secondary oocyte. Only when the sperm cell as well as the liberated secondary oocyte, when they have a chance to meet simultaneously in the ambulatory isthmic junction, there will be a chance or there will be a possibility of fertilization. So here we cannot consider meiosis 2 because meiosis 2 happens only after sperm entry. And also we cannot consider other options in which it is written before liberating polar body. Actually after meiosis 1 there is liberation of polar body. So our answer will be option D here. So remember, meiosis 1 happens inside the tertiary follicle, whereas meiosis 2 happens within the fallopian tube and only when the sperm enters. Question number 5, the phase of menstrual cycle in which corpus luteum produces progesterone from ovary. You know, there are different phases in menstrual cycle. In which phase actually you can see progesterone becoming very high? You might have noticed in that diagram that I have shown, the progesterone level becomes very high during the secretory phase or after ovulation. When the corpus luteum is functional, you can see corpus luteum is producing progesterone, and during the secretory phase or after ovulation, you can see this progesterone becoming very high. So correct answer will be option E here. What about other options? Follicular phase, that is development of follicles. What is ovulatory phase? That is the other name for ovulation. Proliferative phase is the same as follicular phase, but it is with respect to the uterus. We use the term proliferative phase. And menstrual phase, of course, the beginning of the cycle, during the first 3 or 5 days, you can see the menstrual phase. Now coming to question number 6, you have to choose the correct statements regarding oogenesis. 4 statements are given here. You can read them. First one, oogenesis is initiated during embryonic development. That is a correct statement. Second, no more oogonia are formed and added after birth. That is also true. All the oogonia that has to be formed in a female will be there right in the beginning. Before birth itself, all the oogonia will be developed and no more oogonia formed after birth. So that is also a correct statement. What about the third one? Primary oocytes enter meiosis II and get temporarily arrested at metaphase. You know that this statement is wrong. You know why? Because it is not primary oocyte, it is secondary oocyte which enters into meiosis II. Actually, primary oocytes, they have to undergo meiosis I. Then only there will be meiosis II. So the third statement is wrong. It is not the primary oocyte, but rather it is the secondary oocyte undergoing meiosis II. Now what about the fourth one? Formation of primary oocytes begin only after attaining puberty. This is also wrong. All the primary oocytes that has to be formed will be there already before the puberty. As you know, there will be around 60,000 to 80,000 primary oocytes or primary follicles left in each ovary even after the follicular atresia or even after the degeneration of follicles. There will be 60,000 to 80,000 primary follicles left in each ovary or primary oocytes left in each ovary. So this is a wrong statement. So third statement and fourth statement, you can see that they are wrong. So here our answer will be option A. Statement 1 and 2 are correct. All others are wrong. Coming to question number 7, in spermatogenesis, which one of the following is produced after the first meiotic division of spermatogonia? Here we have a set of options, primary spermatocytes, secondary spermatocytes, spermatids, both A and B, and option is spermatozoa. Can you find answer here? You know, which cell undergoes first meiotic division in spermatogenesis? Do you know that cell? It is the primary spermatocyte. So here, after the first meiotic division, there will be formation of the secondary spermatocyte. So the correct answer will be option B here. Okay. Now coming to question number 8. The limited period of sexual receptivity that occurs around the time of ovulation in non-primate females is known as, in non-primate females it is estrous cycle, whereas in primates it is menstrual cycle. So you can pick from options. It is very easy. It is option B, estrous cycle. Luteinization, it is formation of the corpus luteum. Capacitation, that is activation of the sperm cell. It will happen in the reproductive tract. And anistrum is a phase during which the female is not at all receptive, or it is not ready for mating. It is in the case of non-primates. So here answer will be estrous cycle or estrous phase. Answer B. Now here we have the last question of this session. Select the correct option indicating days of menstrual cycle corresponding to FSH and LH levels. In this diagram you can see the different events are shown. What is A to B? Is it follicular phase? No. Yes, it is menstrual phase. From B to C, what is that? It is follicular phase, and from C to D, that is luteal phase. So it is very clear from the diagram. Our answer will be option E here. A to B menstruation, B to C follicular phase, and C to D luteal phase. So with this question, we conclude our session, the second session on human reproduction. So in this session, we covered the topics of spermatogenesis, oogenesis, and also menstrual cycle in detail. Thank you.